All right. Now we are going to look at the second step of our cellular communication process, which is called the transduction phase. We talked last time about the reception phase when the ligand attaches to the cell surface receptor. But now we're going to talk about how that message gets from the receptor on the surface to the response, where something's going to happen because of it in the cell. And in this stage, the message is relayed through a sequence, a cascade of molecules called second messengers, and it's converted into a form that the cell can do something about it. And as it's passed from messenger to messenger, from molecule to molecule, it increases in magnitude. The signal is transferred to an increasing number of molecules so that the strength of that signal can exponentially grow. So in the transduction step, this message is converted into a form that can bring about a specific cellular response, whether it is increasing or decreasing the transcription of DNA and creating proteins, or whether it is a signal to the cell can should start to grow to divide, or whether it is an apoptotic signal and a signal telling the cell to go through programmed cell death. All of those are different examples of specific responses. So one signal activated receptor in this transduction step might activate another protein inside of the cell to carry the message on, and that activated protein will activate another, and another, and another, and another, and another, on down the line in a cascade and a domino, an ever-growing domino-like effect. Now, these proteins that are activated one after the other are called second messengers and are relay molecules. Right, a second messenger is the relay molecules that are carrying the message throughout the cell. But like the primary messenger, the first messenger would be that ligand that bound to the receptor. Now these are the second messengers. You'll see this molecule called CAMP or cyclic adenosine monophosphate, used as a example of a second messenger. It's a very common, very important second messenger that helps in the relay of many different messages inside types, all different types of cells. Um, Now, second messengers, these proteins that carry the signal on inside of the cell, are usually existing inside of the cell constantly, but they aren't continually carrying on the signal because they can only carry on a specific signal when they're activated. And usually they are activated when they are phosphorylated or when a phosphate is added to them. So here is a picture of a generic transduction path step. So let's orient ourselves here on the top. Here's a signaling molecule, a ligand, and here's the receptor. And here on the bottom, this is the cellular response. That's the response phase. So everything in the middle is this transduction step. What happens in this generalized pathway, which is also called a phosphorylation cascade, like you'll see in a second, is that this receptor protein, the internal domain, the intracellular domain inside this part right here, will change shape. It will go through a conformational change when the ligand binds to it. This conformational change will activate one of the second messengers, our initial relay molecule. Hmm? Oops, sorry. And this relay molecule will in turn activate what's called here, it's called an inactive protein kinase. A protein kinase is a class of enzymes that phosphorylate something. It adds a phosphate group to something. So this activated relay molecule activates a protein kinase, right? And this protein kinase works by, when active, removing a molecule of or a phosphate group from ATP, adenosine triphosphate, and sticking that phosphate group on a second molecule. Well, in this case, this second molecule is inactive protein kinase 2. And when that phosphate group is stuck on the second molecule, or phosphorylates it, it activates it. So here, this inactive protein kinase becomes active when protein kinase 1 phosphorylates it. And then works to phosphorylate protein kinase 3 that becomes active, and on and on and on and on and on down the line until we get to this last step, where our final active protein that's been phosphorylated can achieve some cellular response. So it's a big game, it's a big domino chain. It's a big game of telephone. The first one activates, the second one activates, the third one activates, the fourth one on down the line where the final one can activate some cellular response. And so this cascade, if you will, of one protein kinase phosphorylating the next is where we get the term phosphorylation cascade. All right, phosphorylation cascade. Now, this, you might ask, do we have all these steps? Why doesn't the first one just travel into the cell and achieve the cellular response? Well, one main thing. One, or a couple main things. One, this first active relay molecule will never be able to stay active the whole time to go all the way to the cellular response. So it needs to have some sort of way to pass that on, because it is not able to, in its limited time of being active, to carry it all the way to the destination. Secondly, we want the signal to grow. So one signaling molecule doesn't just, for example, cause the creation of one product. We want to make that signal as big as we can, because if we have many different molecules carrying the signal, it will more assuredly reach its destination in a quick amount of time. So and the signal grows because each activated protein kinase will stay active for either a specific amount of time until the phosphate falls back off, or more likely, will stay active for a certain amount of time until a separate enzyme, these purple ones called protein phosphatases, removes the phosphate group from the protein, making it inactive again. So once this protein kinase is active, it will stay active until maybe a protein phosphatase binds to it and removes the phosphate group, and the process starts over again. So when each of these protein kinases are active, they will continually use ATP and the phosphate group attached to phosphorylate a number of different protein kinase 2s, for example. So this active protein kinase 1 might be able to phosphorylate 5 inactive protein kinase 2s before it is dephosphorylated by a protein phosphatase. So then each of those 5 active protein kinase 2s might likewise have enough time to phosphorylate 5 each protein kinase 3s, because before their phosphate group is removed by a protein phosphatase. And so on. Each of those protein kinase 3s might phosphorylate 5 on down the line. So you see the signal is exponentially growing at each step. And that is magnifying the number of molecules are carrying the signal and the likelihood that it's going to reach the cellular response in an exponent way. After that signal has traveled to the area of response, a response will occur. So here is the response phase. So reception phase, transduction phase. Here, look, the last step in our transduction pathway, we might our last relay molecule might travel through the nuclear envelope, through a nuclear pore, and activate a transcription factor. And a transcription factor helps with the transcription of DNA, which needs to occur in order to make a protein. So a lot of the times, in many cases, the end result of a signaling pathway, the response might be to increase transcription and translation of DNA into proteins, or maybe to limit or decrease the rate or the production of proteins from a specific transcript. And so a lot of cellular responses are transcription related, but not all of them. Some other cellular responses might be to create new enzymes, or to rearrange the cytoskeleton, or things like that. But many other times it's upward regulating or downward regulating transcription, and consequently translation and making of proteins. So, like we said, there are many different possible responses. In this example, a response of upregulating, of increasing transcription, ultimately to make a specific protein is the response. Our last signaling molecule in our cascade goes into the cell, the nucleus, into the nucleus through nuclear pore, and activates a transcription factor, which is a molecule that facilitates the transcription of DNA into mRNA. So, like we talked about originally last lesson with signaling molecules, there is a high specificity of this cell signaling process. Particular proteins that a cell possesses determines which signaling molecules it will respond to and how it will respond to them. So, for example, if this red molecule is epinephrine, for example, only the cells that have this epinephrine receptor will be able to respond to the epinephrine signal. And each cell that has this epinephrine signaling molecule might have a different intracellular domain so that a different response happens from that same signal. So here we have one signaling molecule attaching to one receptor and one response happening. In this example, we have one signaling molecule attaching to one receptor, but this second messenger is able to activate two different types of relay molecules, so two different responses happen because of the same signaling molecule. And here we have, like in this one down here, we have a different receptor responding to the same signaling molecule, so a different response happens. Okay, so liver cells and heart cells, for example, do not respond to the same way to epinephrine because they have different collections of proteins. They have different receptors on their surfaces. Both of them might respond to epinephrine signaling, but they might respond to them in different ways because the intracellular domain is different and so might activate different relay molecules. Right. That's going to be it for today. Um, I hope you focus on this and take this into class because our work on the case of Elizabeth Hughes will utilize this information to a really great extent.